Hi, I'm David Summers. Thanks for the opportunity to speak about my lab's research on connectome fingerprinting and our efforts to extend this technique to neurosurgical patients. Connectome fingerprinting combines neuroimaging and machine learning to make fine-scaled, individualized predictions of functional brain organization in humans. Potential applications range from developing highly efficient functional localizers for cognitive neuroscience to non-invasively assaying a patient's functional brain organization for precision medicine. Connectome fingerprinting uses measurements of brain connectivity to predict high-resolution maps of brain function. Dick Passingham conjectured that each cortical area has a unique pattern of connectivity to other brain areas, and that if we could establish these connectivity profiles and measure the connectivity of a bit of brain tissue, that we could predict the functional identity of that tissue. Connectome fingerprinting builds on this idea by applying non-invasive neuroimaging techniques and by greatly expanding the spatial extent of predictions. To build a connectome fingerprint model, we first select a cortical parcellation scheme, similar to what Chantal showed in her talk, of P parcels, and then we select a search space at higher resolution of V voxels or vertices for which we wish to make our activation predictions. We then acquire two types of data on a set of training subjects: functional MRI data of the task contrast of interest and connectivity data. We compute each subject's functional connectome from their resting state fMRI by performing Pearson correlations between the time courses for each vertex in the search space and each cortical parcel. We combine functional connectomes and activity maps for a pool of training subjects and construct a connectome fingerprint model by using ridge regression or machine learning approach. To make predictions for a novel subject, we only need that individual's functional connectome. That is, we only need resting state data to predict their task activation. By applying the individual's connectome to the model yields this prediction. And if we have the actual task data for that subject, we can also validate our predictions. So this is what the model predicts. This is the actual subject's task activation, and that the subjects that we're showing here were not part of the training set. They're separate test subjects that are used here. We've been successful in making highly accurate predictions for attention and working memory activations in frontal and parietal lobes. Predictions are highly individualized. The subject's own functional connectome almost always makes much more accurate predictions than using another's connectome or a group average. Prediction accuracy often matches the correlations between two sets of functional localizers. This suggests that connectome fingerprinting could be used to substitute for task-based functional localization. This offers potential utility in clinical settings for which we wish to precisely map a patient's individual brain organization, but where the hospital lacks a dedicated fMRI team and or the patient is unable to perform the tasks inside the scanner. Excellent work by my PhD student, Vipin Tripathi, in collaboration with Dr. Alex Golby and her team at the Brigham, examines the feasibility of applying connectome fingerprinting in glioma patients. Our long-term goal is to develop connectome fingerprint methods that could be applied across multiple neurosurgical centers, and that will integrate with the neuronavigation systems used in surgical planning and in the operating room. We've used motor task data from the Human Connectome Project to construct models and applied those models across scanner environments to healthy controls in glioma patients from existing data collected by Dr. Golby's team at the Brigham. Here we see connectome fingerprint predictions for a healthy control and a glioma patient along with their actual activations. Despite having a mere 4 minutes of resting state data per subject, we've had enough success to demonstrate the feasibility of this approach. These results are encouraging, but we also see many ways to improve connectome fingerprint model performance, including collecting more resting state data and probing neurovascular coupling. probing the nerve, the decoupling around the tumor sites. We see a potential market here as well. And I'd just like to end by thanking my lab members, past and present, for their excellent work, and the funding agencies, and thank you for your attention. Thank you.